Okay, they're the highest. If you're the highest actually is in the Caribbean, so you look at Jamaica, you see really high rates of prostate cancer mortality. But if you look at sub-Saharan Africa, mortality rates are very high. Okay, I'm not sure what's up with Turkey or Estonia. They're a little bit higher this year. You look at different years, you'll see a little bit different noisiness to the data. Okay, and the question is, what could be contributing to this? Okay, I'm a geneticist, so I'm thinking, okay, maybe there's genetic risk factors, but it could just be access to care. So one thing we could do is we could look in the Southeast United States, we could look on a county level, and we could see, okay, how common is prostate cancer? But then also what we could do is we could base, or how high is prostate cancer mortality? We could also look at per county, what's the proportion of individuals that are African American? And just looking at this, you're like, wow, there's an amazing match between these two. Okay, there's actually one key exception, and I'll get to that in a minute, but this suggests that maybe there's something going on. But you could say maybe it just is access to care. Well, if it just is access to care, and if African-American men are not getting to the clinic that often, then you would probably expect to see the same thing for testicular cancer. Okay, sex-specific doesn't match. Okay, so the question is what's going on? And maybe there are genetic factors. The one exception to this is actually where I live in Atlanta. And we actually don't see a high prostate cancer mortality in Atlanta. And some of that might have to do to Emory, Piedmont, Grady. These hospitals are doing a pretty good job of screening in Atlanta as opposed to the rural South. So even though I'm going to be focusing on genetics, you have to be a little bit cautious in the sense of thinking like, well, there are plenty of other factors that play into this. Okay. So one thing to keep in mind is in the United States, the mortality rates of prostate cancer are roughly 2 to 2.5 times as large if somebody is of African-American descent. So a number of years ago, collaborating with Matt and with Sarah, we looked at what the Tishkov lab calls the 1M data set. And what we did is we looked at predicted risk of prostate cancer through many different ancestries. We looked at 1,000 genomes populations, and then the 4-letter codes or Tishkov lab data sets. Okay, and so when we did this, what we see is that the populations with the higher predicted risk of prostate cancer, knowing what we knew in 2018, actually tended to be West African. They tended to have this orange and maroon ancestry color here. And one thing you could do is you could say, well, what's this polygenic risk score? Let's get how you scale it. Can give you a little bit different answers, but then you can see that the more orange that somebody has, the higher predicted risk of prostate cancer. And then way down here, the more pink or gray, the lower predicted risk. This actually matches mortality and incidence rates when you look across the world, because prostate cancer rates are actually lowest in East Asia, highest in populations of African descent. Okay, so this suggests that there might be some heterogeneity for prostate cancer, that maybe West African populations have a little bit higher risk. But the thing is, when we look at these, at this time, we look at roughly 65 to 68 different SNPs that are associated with prostate cancer. What we then did is said, well, are any of these under selection? And the vast majority of these are in regions that are just evolving neutrally. They're just like the rest of the genome. But one region had a really interesting pattern. Okay, so this is looking at Sabeti's CMS score. So it's integrating a number of different scans of selection and just reading the y-axis. A high score means that there's greater evidence of selection. And you can see that there's this relatively large haplotype that seems to be under selection, or at least have gone through some sort of a sweep in Europe. Okay, we don't see evidence of any sort of selection in Africa or East Asia when we looked at this. It's got 2 genes here. One is related to hormones of prostate cancer, PRLH, but right next to it is MLPH, okay, which is a gene that's associated with pigmentation. So it's known that in dogs, if you happen to have an MLPH mutation, basically it leads to dilute color. Okay, so in humans, you can imagine it's leading to dilution of whatever pigmentation that individual has. And then there's multiple papers that have looked at, okay, vitamin D, natural selection for pigmentation in different parts of the world is sort of consistent with maybe this MLPH haplotype selected in Europe. And it drags along a protective allele for prostate cancer that makes it so that Europeans have a little bit less risk of prostate cancer. It's not a big effect of it. We're talking about an odds ratio of like 1.16. But the idea that when we're looking at differences between population, we have to also just think about what's in that DNA right around those alleles are associated with disease. Okay. So then what we could do is we could think about, well, what do we know about disease risk right now? Okay. So we could think about how things might be biased. So everybody's familiar with the GWAS catalog. And as of last week, there were 7,000 studies and 692,000 associations. Okay. Some of these are redundant, right? So you have a SNP that's associated with 15 different traits, or maybe it's 10 of the same trait, 5 others, so there's polytropy going around. But there's a huge amount of these associations that are there. And hopefully we all know how GWAS work. But then the question is, what can we do with all this GWAS information? So I try not to listen to conversations in bathrooms, but I was at an American Society of Human Genetics meeting at least like 8 years ago. Two guys walk in and they're talking to each other. They're like, we're all post-GWAS. We've got to move on to SPWAS now, you know. But we've been thinking about like, okay, GWAS ends up being able to conduct genome-wide association studies is really useful, but we need to move beyond that. What can we do to actually understand traits? And as students think about the functional genetics of this, you know, how do we map things to causal loci? But one thing you could do is you could use our findings from GWAS to try to say, okay, can we predict traits? Can we generate these polygenic risk scores? Simple way to do it, you're just counting up how many, the dosage of an allele, 0, 1, 2 copies, you're weighting by effect size. And so this paper by Kerr et al. Is looking at coronary artery disease, and you can see that individuals in the very top percentile of risk have a much higher prevalence of coronary artery disease. But there's still some individuals in the lowest percentiles that still get this disease, okay? So you can think about this as basically bidding individuals in terms of their predicted risk, okay? But the thing is whenever we're looking at polygenic risk scores, we're aware that allele frequencies differ across populations. Biggest issue is also if it's a private allele. So what if there's an allele that influences risk for a trait, polymorphic in that discovery population, but guess what? In another population, everybody either has the protected allele or everybody has the risk reducing. Okay. So the other thing is that we can also think about other things that might differ. So effect sizes can potentially differ across population. This is an ongoing debate. So somebody like Bogdan might be saying, oh, no, effect sizes are the same across the board. Somebody like Elizabeth Atkinson might argue that, you know what, maybe they do. Maybe they do vary by ancestry. Okay. The other thing is we can also think that the ability to tag variants can actually matter because often GWAS or polygenic risk scores aren't looking at the causal allele. They're looking at a tag set that's basically representing that haplotype, but that could actually lead to some problem, especially when LD differs across populations. So what we did a few years ago is we looked at a set of roughly 3,000 independent GWAS hits. All of these are associated with some sort of disease, so they're not associated with height or I'm not going bald, or I am bald, but I'm not calling this disease. And my lab's like, yeah, right, Joe. Okay, but basically these are disease-associated alleles. And the first thing we did, we just said, what's the frequency in Africa versus outside of Africa? Okay, and pretend you're colorblind, completely colorblind. Don't look at the colors just now. Okay, just look at the points. And what we see is there's this excess of points in the bottom left-hand side on this along the y-axis and along the top right. Okay, it's not symmetrical. Okay, and the reason for that is that a lot of these studies are done with populations of European ancestry. Some of East Asian. So what we're doing is we're picking up these, but we're not picking up these as much. They basically have to be polymorphic where the studies are done. Because I'm an evolutionary geneticist, I basically was like, well, wait, a really boring plot if we just put gray circles. So let's color them by ancestral derived state. Did not really expect to see something this striking. And so when we colored by whether the risk increasing allele is ancestral, so shared with chimpanzees and other primates, or derived due to new mutations, something in the last 6 million years, we get another asymmetry. So there's more blue up here and more red to this side. And it's sort of a very perplexing thing. We're like, why should there be any bias? I mean, we're all equally related to chimpanzees, okay? There's no reason, at least initially, why we would expect to see that. But it turns out that there's ways you can sort of pull it apart. And hopefully this sort of thought experiment will make some sense. So just imagine we've got something prior to subset of humanity living in Africa. We're only tracking 2 alleles right now. We've got this low frequency allele that happens to be, in this case, that's a derived allele in blue, and then the red is the ancestral allele, and it's equal frequencies in both places, okay? And then what we can imagine is you've got genetic drift. We also have stronger amounts of drift outside of Africa. So you've got this out of Africa, bottleneck and founder effects occurring. So you think about diffusion of these, just like that simulation I showed, there's more movement on that y-axis than there is on the x-axis. So imagine you've got a lot of SNPs that start out in that blue position. You get movement left to right, but most of it's vertical. Same thing with the red, okay? Now we have to think about where were the studies done. So if you do a study in, say, Europe, you're not going to be able to catch stuff where the minor allele frequencies are really close to 0. Okay, you're not going to catch something if it's got an allele frequency 0. Of course, you can't detect it. One, you can't detect it. Your power to detect it is actually maximizing this middle band. So what's going to happen is you'll detect this guy, but you're not going to detect this other one down there. Okay, so what it means is that you sort of, the veil of detection is basically going to mean you're going to have excess of blue here because you don't see those. Same thing with red here. Now, if you did this study in Africa, it's going to be a little bit different. You're going to detect both. You're going to lose the stuff on the far edge. But the thing is that some sense in African studies align to the direction of drift. And Matt gave me rolled his eyes, so I said, it's like polarized light. And so I started thinking about that, but it's almost like, where has there been more drift in allele frequency, and then where's the study done? Okay. And so we did a number of simulations looking at this to try to look deeper at it. But this is one possible explanation for that ancestral derived asymmetry. You might be able to come up with other ideas too, but this satisfied us at least when we did. There's other biases that come into it, and this has to do with genotyping technology, right? So often you're not well funded. Most people are not well funded enough to do whole genome sequencing of everything. Now we've got good imputation panels like TopMed, but basically, if you're using a genotyping array, you have to think about where were those SNPs identified. So those SNPs were identified originally in a population of European descent. Well, they're going to be really good for European polymorphisms, and you're going to detect stuff that have to do with European polymorphisms. So that same bias I was showing before, it's not just where the study's done, but it's also where are the SNPs that you're looking at, where were they originally ascertained, okay? So it hints that if you're trying to do a study, you want to have an array that basically does a really good job of looking at, of tagging polymorphisms in your population of interest. You can also think about what ancestries the studies are done. I love these photos from National Geographic. They're from, I think, Queens, New York. I think when I think of like, what are some of the most admixed neighborhoods in the US, everybody in that panel basically is checking multiple boxes on a US census. We're sort of headed there as a species. But one thing we could think about, and I was tempted to use Alicia Martin's plot here, but this actually is a little bit more updated. And the idea here is we could say, okay, what sort of ancestries have recent GWASs looked at? And we still see this massive bias towards European ancestry studies. Okay. Part of the problem with that is that UK Biobank is a great tool. So a lot of people use UK Biobank. And guess what? We just keep on being biased that way. So most of what we know about disease genetics happens to come from your European ancestry populations, which is one reason why there's this real need to look at populations that have diverse non-European ancestries. And so for the next few minutes, I'm going to talk about 3 examples of how genetic predictions that are based on SNPs that were ascertained in largely Eurasian populations, why they might not generalize to Africa. And I'm going to look at height, baldness, and prostate cancer. So this is related to the study that Matt and I are hammering out this week. And so one thing we can do is we can look at how well the predictions of height generalize to other places. So you can think about it, if I had a set of SNPs that were ascertained in European populations, could they predict that Shaquille O'Neal is way taller than Kevin Hart? Incidentally, he owns the Papa John's pizza half a block from where I work. And Atlanta has different celebrity sightings than Philadelphia. Okay, so we're saying a Tishkoff lab data set here, which is a combination of the 1M and the 5M data set. And what we're doing is actually saying, how well do these predictors of height generalize to Africa? So there's a polygenic prediction from a few years ago from Lilo et al. It's based on a machine learning approach, appeared in the journal Genetics. It's by Stephen Sue's team, and I'm very skeptical of it. They basically, I think they're overstating how well it works. When we apply it to African populations, these are really bad correlations. That's the R, not the R squared. So you square that and it's just about nothing. Look at this correlation between actual height Z-score versus predicted height. It's very, very weak. If you're an optimist, you're like, yeah, the slope's positive, but that's asking a little too much. Then what we could do is say, okay, what if we looked at a better predictor of height? So one of the best GWAS of height out there looks at roughly 5 million individuals. So you've got huge sample size, but once again, it's biased towards European individuals. And we can see that it's still really noisy. Okay, each data point here is a separate individual from the Tishkoff lab data set, and it's color-coded by their primary ancestry group. Okay, and better correlations, still positive, so it's doing some job predicting, but it's not doing that great of a job predicting. Okay, but we can also look in other ways. And this actually, I think, might have come originally from Matt years ago. This idea of like, let's not just look at that. Let's compare it to if you knew the ancestry proportions of each individual. So you've run admixture, and you've identified, okay, this person is 70% light blue, 20% yellow, 10% green, and you knew the average height of each of those different ancestry groups, you could generate some sort of a proxy for height. And the idea is that that's catching some of these rare variants that maybe you don't have statistical significance, but you're catching something about the genetics there. What happens with this is it actually does a better job predicting. Okay, and these are complementary approaches. One is using a huge GWAS of largely non-African samples. One is just using the ancestry proportions, but they're both able to do some job predicting. It depends on which group you look at. So we've done finer scale analysis where once you look at like the brown points in that plot, the slope might be flatter, but if you look at the light blue, so individuals that are labeled Hadza here, it's actually a pretty good correlation within Hadza. You can see the blue trend is upward in that. So this is basically saying that GWAS from non-African populations for height, even when they're really, really well powered, aren't going to catch it that well. Okay. Then we can look at male pattern baldness. And if you're familiar with AUC statistics, you're like, oh man, that makes me cry. Okay, so this is basically saying, and once again, as we're doing this, I'm losing my hair. But one thing we can do is we looked at two different European ancestry GWASs for male pattern baldness. One was Hagenars, the other one is Hillman Highbox. This is a little bit more continental Europe. That's more UK Biobank. AUC statistics, once again, 0.5 means it's basically as good as chance. It's not really a good predictor at all. An AUC statistic of 1 would be fantastic. And it leads above 0.5, but your confidence intervals are barely there. This one's actually intersecting 0.5. It barely tells us anything about baldness in Africa. Okay. And this is using a data set from the MadCap network. And then what we do is we zoomed a little bit more and say, well, what if we don't just look at the overall distribution of the populations we're looking at, but let's look at the individuals in the middle of the distribution for our polygenic risk scores versus the top 10%. We do that, we actually see that there's it's somewhat informative. Okay, so individuals on the tail end of our polygenic score for baldness seem to basically be enriched for having more hair loss at age 45. Okay, but once again, the predictions aren't generalizing. Okay, and I had this really, what I thought was an awesome hypothesis of like why things are not generalized. And I was like, I realized that like, if you go back to 2010, and actually in subsequent papers too, there's this knowledge that Neanderthal DNA seems to be enriched for, in modern humans, Neanderthal DNA seems to be enriched for genes that are involved in keratin-related phenotypes, you think skin and hair. So maybe it's not generalizing because there's Neanderthal alleles that are predicting height in Europeans, and then these African populations don't have Neanderthal DNA. Maybe that's what we're seeing. I wish. Okay, and so I needed a picture of a bald Neanderthal, and this is actually from the Natural History Museum in Vienna, where Matt Hansen, my wife, and I, we all walked around there right after an SMBE conference. I don't have a picture of us hanging out with this guy, but we actually have seen the statues. And so what we did is we basically said, okay, let's look at these regions of the genome that are associated with baldness. Let's see, do they have more or less Neanderthal DNA than you'd expect by chance? And the way to think about this is the red and the blue are just our Neanderthal, our baldness associated SNPs. The gray curves here are replicates of control SNPs. And the sad truth is that you look at these, it intersects with the gray. So they're not enriched for Neanderthal DNA. So the hypothesis was great, but data does not support it at all. The reviewer, one of the reviewers on the paper was like, yeah, you don't have that much power to detect this. And they're actually pretty right. We have pretty weak power to actually detect this. Okay, so it seems like the poor prediction for baldness isn't just a case of saying, oh, it's Neanderthal DNA. We've actually found some evidence that the genetic architecture of this disease does differ between Africa and Europe. One key difference seems to be an X-linked hit near the androgen receptor, which is at nicely intermediate frequency in Europe, and it's less than 5% in West Africa. Okay, so we see some impact in terms of that. Okay. Then we can turn to prostate cancer. And so once again, we're using the MadCap network data set. I'm going to talk a little bit more about that in a couple minutes. But what we're doing is we're looking at a couple different polygenic risk score sources. So some of these, the Karuna Muni one deals a little bit more with age of onset. The Conti one was at the time the best GWAS related to this. So that's Chris Hyman and David Conti. And first thing we did, and once again, sometimes people are be very cautious when you do this because often it's not going to match the actual incidence or mortality rates. We look at different global populations, we see which populations had the higher predicted risk of prostate cancer. And sometimes when you do this, you'll get something that does not match the actual empirical data at all. But when it comes to prostate cancer, we see that the predicted risk of prostate cancer is higher in people of African descent. This mirrors what we had in the earlier paper. East Asia is a little bit lower, but it's also actually got some errors here because the actual risk of prostate cancer are not, in reality, aren't actually shifted that much. So it still seems to be overestimating the risk of prostate cancer in African populations. But what we can then do is say, well, how well does this predict an African population? So what we did is we looked at different ancestry populations, but we also looked to see what if we use weights for these polygenic scores that are trained on African ancestry individuals, Asian individuals, European ancestry individuals, and see where do they predict best. So what we did is, in this case, we looked to see for the Conti PRS, which once again was generated from it's a multi-ethnic cohort, but it really was a European cohort largely. and you see where does it perform best in the UK biobank. And it does the best job of predicting when it's ancestry matched. So if you have European predictors, makes sense, they're going to work well in a European population. Of the Conti PRS SNPs, when they use the African weights, that actually gave the best prediction in Africa. But you can also compare these columns. You're like, yeah, but it still was a way lower AUC statistic. Slightly better when it comes to be ancestry matched. We also looked at the Karinamuni polygenic score, and what they have is the primary one, but then they added 3 additional SNPs that they ascertained in African Americans. And you can see that doesn't perform as well, but we see that still ancestry matched polygenic scores generally are going to perform better, which makes sense in terms of intuition. Okay. And so given that, we can think about why do these polygenic predictions generalize so poorly? And you might all have your ideas. These are sort of the 3 ones that in any paper on this, if you're like, what do I put in my intro section? I'm like, these are the 3 things that jump to mind. Okay. You might order these different in terms of which you think is most important, but all these can be there. So one is that you could have effect size differences between populations, right? And that could be due to epistasis. It could be due to genotype by environment effects. But the idea is that maybe the same SNPs are associated with this trait or disease, but just the effect size varies by ancestry or by sampling location. The other thing is there could be LD differences, right? So one thing you could have is like, oh, we're tagging it, we've got this region of the genome that's associated with a trait. The original study was done in Japan. You're tagging a 90 kb window, and your lead SNP is on the far right of that. Maybe the actual causal SNP is far to the left. And because you have different LD structures in Africa, maybe you're actually not doing a good job of tagging the causal allele if you use that lead SNP. Could work as well. The other thing is allele frequency differences, okay? So if you have an allele frequency of like 40% in one population, 1% in the other, you've only got, you're lacking statistical power to replicate that where the allele is rare, okay? So these allele frequency differences might also matter. So a priori, these are sort of 3 reasons why you might see something different. And so what we wanted to do is actually look at that, but we wanted to look deeper in terms of genetic architecture. And so one thing we did is we dug much more deeper, and this time we're really focusing on the trait of prostate cancer. We're looking in sub-Saharan Africa. And to do this, you need a decent sample size. So one thing that we did is we worked with other members of the MadCat network. So Tim Rebick was at Penn. He left right around the same time I did. He moved up north. But this consortium really got rolling about 10 years ago. And as you can see, we've got a large number of African centers. The primary hospitals that are here, there's 1 in Senegal, there's 2 in Ghana, 2 in Nigeria, and 2 in South Africa. But there's other teams that are slowly coming online, which is that we're still applying for grants to get funding to do additional genotyping at their sites. Also, a number of different sites in the United States. And there's a couple main reasons why this consortium was founded. First thing is we want to be able to build research capacity. So what makes this study actually really cool is that when we look at prostate cancer in Africa, it was the first major GWAS where over half the samples were genotyped in African soil. Okay, so it's not just a case of, okay, send the blood back to the United States, genotype them. We're really trying to integrate. Also, the team has clinicians, we've got epidemiologists, we've got, bioinformaticians over there. It's a really great community. Okay, but really what we want to do is we want to say, well, let's look where this disease has the biggest health burden. Let's see, can we detect novel loci? Can we detect, alleles that maybe we're missing when we're only looking at European ancestry populations or admixed ones in the Americas. And when we started this, we realized that, well, first of all, we didn't have enough money to do the whole genome sequencing of thousands and thousands of samples. And then we realized, like, you know what, there's not a great genotyping array for where we're looking at. So there was the H3F array, which was being slowly developed. And we're like, okay, that's not optimized for cancer. Also, when it comes to, then basically there's the Encore array, but the Encore array has its own problems because it's got a very high density of SNPs in known cancer regions. So we need something that was basically better genome wide, but then also more focused on Africa. So we developed what we call the MadCap array. We worked with Affymetrix and Thermo Fisher, generated a custom array, and never do this if you have an opportunity. It's a lot more work than you think. But basically we added the SNPs that we wanted, right? So we wanted to have, if it was a known cancer locus, we wanted to have that pretty high density in terms of SNPs. We needed a good GWAS backbone. We're studying prostate cancer. Every single known EPGL that was associated with the disease, boom, put it on the array. And then also we wanted to have good overlap with some other arrays, okay? So this actually allowed us to do a good study. You might be able to get away with it if you're using the Illumina 5M because it's got a good density of SNPs throughout the entire genome. Okay. Also, as imputation panels get better, your choice of array might matter a little bit less. Okay. So we use this technology. And then ultimately what we did is we combined the MADCAP data set with the data set from Uganda, from Practical Consortium. And so what we're dealing with is roughly 7,400 and change samples. Okay, which if you compare it to a major study in the US, you're like, oh no, it's not 250,000 people in all of us. But in terms of the Africa GWAS, this is actually the first, in some sense, I say Pan-African, even though we're only dealing with 5 countries here, but it's the largest study of its kind that's been done in Africa. Okay. And one thing that was nice about this is that these are relatively matched. They're age matched and sample accrual. We had really good screening things. We also integrated a little bit with the NIH had a Ghana prostate study that was a little bit older. And that led to all sorts of QC fun because you had individuals that showed up to the clinic 10 years before the MADCAP study, and then they magically showed up again for a different study. And when we eliminated individuals that were related, we did it within study, and then we realized we had clones, but they were the same individuals that had come through different studies. So rerunning a GWAS, exact same GWAS, 7,450 people, eliminating like 10 people and rerunning the exact same thing all over again and getting basically the same results. Okay, so all sorts of technical challenges, and it wouldn't be a GWAS without a Manhattan plot. And I'm like, yay, there's 3 clean peaks. Okay, so we've got these major peaks, and AQ24 is a region that's really known to be associated with prostate cancer. It's got a lot of heterogeneity there. And it's actually pretty close to MIC. So it makes sense. And so even though the GWAS hits are right next to PCAT1, I've had conversations with some people in this room about like, yeah, what genes are really being affected? Probably MIC's what's mattering. But you can zoom in on some of these core hits we have. So we got 15 prime mapped ones that meet a traditional GWAS threshold. And then we have these LD pruning, and we have 90 of them that are independent genome wide. And we look at this, we sometimes have ones that look like this, which are really interesting, where the allele frequency for this is roughly 25% in Africa. It's basically 0 outside of Africa. So you do a study in, say, in India, you're not going to detect this, even though it might have a decent effect size. What's nice about this too is that it replicates in different parts of Africa. Okay, so we see that cases have a higher frequency of the risk increasing allele. Okay, so and actually a decent number of our lead SNPs actually had that sort of profile. So we're catching stuff that you might not catch if you didn't look at Africa. And so then we want to think about, let's get back to this question of why do predictions not generalize across populations? So what we did here is we said, okay, we could look at the time, the best study of prostate cancer that was non-African focused. Okay, so Enki Wang's paper, they did add some African ancestry data sets, but it's basically the next step from Chris Hyman's group of looking at a multi-ancestry cohort. And we look at their top roughly 450 SNPs. And we said, okay, let's get the ones where we have genotype information. Let's see, do they have decent p-values in our study or not? Okay, and in this case, we're using a very liberal p-value threshold. We just said, is the p-value in our study less than 0.05 or greater? Okay, and we actually saw that we had enrichment for some of these. But what we want to figure out was why. Why do some of these associations replicate across population and others do not? So the first thing you could do is look at effect size. And if they had a big effect size in the Wang et al. Paper, turns out that they're more likely to replicate in our African study. Makes sense. And this SNP is associated with a bigger effect on prostate cancer risk, more likely to replicate. There still were some other ones that had actually pretty small effect sizes that did not replicate in our study. Okay, so that fits with one of the 3 possible causes. The other 2, I expected to actually see that LD differences would make a huge difference. I expected minor allele frequencies to make a big difference. We looked at LD in multiple ways. We looked at recombination rates, we looked at the ratio, we looked at the difference, but we really didn't see any difference in the blue and the gray when we looked at LD, okay? Which is sort of weird. So it does, at least for this set of SNPs, the ability to tag the lead SNP doesn't seem to matter as much as I would have thought. Now, you need to be really cautious here because this is prostate cancer is one trait. We need to look at it in terms of a much larger study. When we looked at minor allele frequency, we would have expected the ones that replicate are going to be higher frequency in Africa, and the ones that don't could be things where maybe the frequency is at 1% in Africa, maybe 20% in Europe. We actually don't see much of a trend there. I'd be a little bit more cautious about this, but the idea is that the allele frequency difference sort of explanation, it's not a big smoking gun here in terms of why things replicate or not. We're expanding this to look at blood traits, and actually it makes me feel like I would be very cautious we're actually seeing something where actually it is a little bit lower, okay? But it's important to actually look at the data and say, okay, a priori we've got these explanations, but what does the data actually tell us about these? So as everybody in this room is aware, Africa is not monolithic. There's a lot of population structure, right? So when we did this, we didn't want to just say, oh, Africa versus Europe. We wanted to say, no, let's look at different parts of Africa. And given our study design, we could divide our data set into East, West, and South Africa. And what we did is we looked at our top 90 associations for prostate cancer. We wanted to say basically how much of the observed heritability is in each one of these 90 lead SNPs. Okay, these are using a marginal significance, so 10 to the minus 5. And what we did is we looked at the genetic variance. So one thing you could do is you can see the GBC is roughly 2 beta squared P1 minus P. And when you do that, the problem is that sometimes it's a little bit different for each region. So we looked at the proportion of these. So if you add up each column, it adds up to 100%. Okay, so basically saying that, does this SNP make an outside proportion in this population versus another? Okay, and these are X-linked variants, so it actually seems that X-linked variants contribute more to prostate cancer risk proportionally in East Africa than West. You can see we've got these number of different independent hits in AQ24, but it sort of hints that, relatively speaking, our South Africa data set, AQ24 matters, but not as much as it does in the West and the East. Okay, and when we think about this, you can think about like, okay, we're representing, and this is actually a major challenge, and I think it's an ongoing challenge of like, how do we represent the genetic architecture of a trait in different populations? There's no perfect way to do it. And one way that we thought about this is that we can represent a population's genetic architecture as a point in a 90-dimensional space. Okay, it's actually one less dimension because you've got degrees of freedom there, but basically you can think about, well, the first dimension is what proportion of the variance is here, next dimension. Some of us might be able to envision 3-dimensional, 4-dimensional spaces, but 90, good luck. Okay, so this is a 2D MPS plot that's condensing that space onto just 2 dimensions. Okay, so reducing it there. And what we have now is we initially had it with a single point for West, East, and South. And we're like, oh, wow, they're far apart. Great. But we've got a lot of uncertainty, right? So we have uncertainty in estimating allele frequency. We have uncertainty in estimating effect size. So what we did is we used a resampling approach where we generated those vectors over and over and over again to get this cloud of points and saying, okay, the genetic architecture, in this case of South Africa, is somewhere in this region. Okay, so we've got and basically we're saying, is there any overlap between these clouds of points? Turns out that there isn't, at least for this trait. Okay, and you might be wondering like why this is a lot tighter than here. Well, this is because we have bigger sample sizes in South Africa. Our East African data set is actually a bit smaller. That's why there's a lot more noise in terms of that. So if you were doing a study, we only had 100 individuals for a region, your cloud of points might be covering a huge amount of space. So one way to think about this is you probably need a few thousand individuals, at least with this method, to be able to distinguish these. Okay. But there might be other ways that you could represent the genetic architecture as well. And so when we see these, we see that there's these non-overlapping clouds. We want to think about, well, why they might be different between West, East, and South Africa. And one way that we could think about this is say, well, what about allele frequencies? The other way is thinking about effect size, because that's what we're using to generate these statistics. I'd like to think I know African population genetics. If you had asked me to like plot West versus South allele frequencies for the 90 SNPs we had here, I would have expected a bigger spread. It's actually pretty poorly. Okay. And so, but what we see is that for allele frequencies, it's close to the diagonal. So yes, there's allele frequency differences, but we're not seeing cases where it's like 60% in one part of Africa and 5% in another. Okay, there might be a little bit of bias here because we're looking at stuff that came out of our meta-analysis of all these regions, so we're less likely to catch those. But when we look at effect sizes, we see that there's a lot more noise. Okay, and this is even more forgiving because if you basically made a 95% confidence interval cross, you see huge crosses for each of these. Okay, and basically what that means is that our allele frequencies are much more similar across populations. Our effect sizes are not, but we're also a little bit worried because we're like, well, our ability to estimate effect sizes is a lot more limited, right? So to get a good allele frequency estimate, if you have 1,000 samples, you can get a pretty good allele frequency estimate. If you have 1,000 samples, it's actually pretty hard to get a narrow effect sizes. So what we did was we used the same sort of approach I described before where we had these cloud of points. But what we did is we said, what if we make it, we're going to hold the allele frequency constant. So the only thing that varies is the effect size. Now we'll see how far apart these things are. And then we did the opposite. We said, we're going to hold effect size as the constant. So basically use our meta-analysis effect size and then only vary the allele frequency and then see how far apart these clouds of points are. When we did this, we looked at the Euclidean distance between each of these 3 clouds. And what we see is that when we allow effect size to vary, these clouds of points are farther apart. So at least for this trait, you need to be really careful not to generalize this too much, but at least for this trait, it seems like the effect size differences are driving the genetics differences in the architecture a little bit more than allele frequency, okay? But once again, you have to be a little bit skeptical here. The thing is that we actually see that there's some overlap here too. This is narrower for the allele frequency just because there's less noise when it comes to it, okay? And plenty of other things can be acting here, but we're trying to get a handle of like, yeah, why are different regions of the genome associated with this trait in one part of Africa as opposed to another? And so being a population genetics lab, we wanted to say, okay, let's look at these top 90 hits. What could we see in terms of them? Now we've got a different sort of ascertainment bias. Remember before, there was a lot more on the y-axis. Well, now we did the study in Africa. We get the opposite trend. We've got SNPs that are 10% in Africa, nearly monomorphic outside of Africa, okay? And they're color-coded here so that where the minor low frequency is below 1% in Europe, basically that's going to be gray. Okay, so you see everything on the top and the bottom. So some of these alleles that we found in our African study, actually the African populations have a high frequency of that risk allele. Sometimes they actually have a lower. Okay, there's not necessarily a big bias either way. But what we're doing is we're catching the stuff that you might not otherwise catch. All these ones in blue, these are ones at intermediate frequency that if you did the study anywhere in the world, you probably would catch up. Okay. But then what we wanted to do was say like, okay, what sort of properties do these have? And actually with these, because they're so rare, when they're rare outside of Europe, you can think about two different things that might be acting. One is that they're a new mutation. So maybe it comes in the last 60,000 years. And that's why we don't see it in Africa, right? So it's, subset humanity leaves, and then there's a mutation that in West Africa 30,000 years ago. That's why it's locally found. The other thing is maybe it was lurking at moderately low frequency and it just didn't get out of Africa with the founder effects and bottlenecks. So what we did is we looked at estimates of allele age, and you have to be a little bit cautious here because any estimate of allele age is going to have crazy confidence intervals. If you try to use Jiva or there's a website genome.dating where basically you could look it up. Any method you use to estimate allele age is going to be pretty noisy, whether you use a mutation clock or a recombination clock. But what we found is a lot of these are generations, not years. A lot of these mutations in our study that were associated with prostate cancer are actually pretty old. So they predate the out of Africa migration. That makes sense, but you have to be aware that we have only 7,500 individuals. We don't have the power to detect an association with something that's really, really rare. So we're only looking at alleles that are intermediate frequency, and to get intermediate frequency, they need to have been around for 100,000 years or 50,000 years or 125,000 years. Okay. And so what you can see here is that we do have some of these older ones where the frequency outside of Africa is very low. So that hints that this idea of bottlenecks and founder effects might be contributing to why these are, I'll say private to Africa, but I'm using it in a very loose term. I know Graham Coop had a tweet a year or two ago that's like, oh, no private alleles exist. If you sequence everybody in a population, you find somebody with that. It's like, yeah, but they're not identical by the same. So these are ones that are pretty rare. Okay. The other thing is we wanted to look and see, is there any major evidence of selection with this set? And we basically said, are they enriched for extreme IHS scores? And the null expectation is that they would be normally distributed. These are basically thinking about z-scores. And it looks like what you'd expect if you look at the rest of the genome. Okay. So basically prostate cancer SNPs, even though sometimes they might be linked to things under selection, in general, and this is looking at hard sweep signatures, but when we looked at IHS statistics, they don't look like they're under strong selection. But that's going to make some sense because prostate cancer acts so late in life that even though it might have severe consequences to somebody that gets it at age 50, how much is that affecting an individual's number of offspring, and survival? Okay, so we basically see that overall, it seems like these are evolving neutrally, at least as a collective set. So going forward, you can think about, well, how might we improve genetic predictions? Well, one is conduct your GWAS or conduct your studies in diverse populations. Okay. Even if you don't, be aware where you're conducting it and try to correct for that somehow. Okay. If you're generalizing predictions, try to use ancestry match data. If they're not ancestry matched, maybe you have to somehow correct for it some way. Okay. Maybe that involves leveraging information from evolutionary genetics. So maybe you want to be able to leverage, the fact that the patterns of LD are different. That might matter a little bit if you're trying to translate stuff from one population to another. And the reason why we have to think about this is if you did a study in the UK and you did another study in Japan and you say, I'm going to try to predict a trait in Pakistan, well, there might not be a well-powered study in Pakistan. So you want to be able to say, how do I combine a huge study here, a moderate study here? Neither one's ancestry matched, but I don't want to just say we have no idea about the genetics. Okay, so you want to weight those. And then this is something that, even though I didn't delve too much into it at all in the talk, that functional annotations matter, right? So if you knew that this was basically, if you have a set of SNPs that are associated with a trait, and you know that, like, well, these 30% of them, they're in an androgen receptor transcription factor binding site. It's like those might be actually much more causal. Maybe you want to weight those more when you're generalizing. So hopefully it made some sense. With that, I'd like to thank my lab. This is our current lab. Donovan couldn't make the picture, but yeah, fun bunch of people at Georgia Tech. We're in Midtown Atlanta. Once again, I'd like to thank Ben for giving me a chance to give this informal talk. At first I was like, oh, just a conference room on the 4th floor, but it's nice to see a little bit bigger group of people here, a little bit like a homecoming. This is a picture of Madcap Network in sunny Cape Town. It's one of our nicer dinners, so going out to a vineyard and talking about science and health. But also funding sources, so an IGMS, NCI, and then currently finishing up a training grant from the Department of Education, which is still with us for now. So with that, I'd like to thank you guys for listening. Yeah.
Yeah, nice talk. I have a question about the so you had sort of looked into the heterogeneous genetic architecture within African subpopulations.
Yeah.
And previously you had another slide where you're sort of using admixture fractions to do genomic prediction. Is that admixture fraction computed from this sort of within Africans only, or is it?
Yeah. So when those were different studies of different individuals. So are you talking about this one, or the previous one? Yeah.
You had mentioned this.
Oh, yeah, the height one, maybe. Yeah. So that was a different. Yeah. So when we did this, that's actually a good question, and actually Matt to thank on this. So I do think when we did this, we actually downsampled the data set here, so that we had trained it so that they were not the same individuals. For that, there was a downsampling procedure that was there, because you don't want to be generating your admixture proportions from that and sort of having this double use.
Yeah.
If that makes sense. So I don't know if that directly answers your question.
Well, I guess I was just sort of wondering, the admixture fraction, is it admixture with respect to running admixture within Africans?
Yeah. So it's running it on Africans and then a small number of thousand genomes populations. So it's using GWD, YRI, WK, so using the African ancestry 1,000 genomes populations.
So the way we can understand it is it's sort of capturing maybe a bit of within Africa population structure, in terms of the admixture.
Yeah, let me see if I can try to get at that. And this is from Matt and Sarah's the manuscript we're working on, but let me see. These are figured one, right? Yeah. So it's using these sort of proportions, right? So the idea is that if you knew the average height of if somebody was 100% light green, you could try to predict their height on some sort of z-score scale. And then you knew the average height of somebody that was 100% yellow, 100% pink. And basically you're doing a weighted sum of all these. Okay, and it's a crude measure, right? But it's something where but if you have empirical evidence that this population is taller, the question is how much does it correlate with it? So we actually saw this way back when we started to look at that. This is way back to 2011 in Sarah's lab. If you looked at rainforest hunter-gatherer populations, you look at the same Baka, and you look at proportion of presumably Baka versus, I guess, TCAR South or neighboring farming populations, you see that the more Baka ancestry somebody has, the shorter they are on average. But it's not fully linear. I think some of that is just that there might be health consequences of being too short. You can think about that sort of thing, but you see this gradual trend even within the same study site in terms of that. So you can zoom in and say, even among Hadza, you see that, these are sampled from same locations in Tanzania, but the idea is the ones that have more, so whatever this light blue ancestry is, tend to be a little bit shorter. Okay, so we do see at least some evidence of that, at least for those populations. For some of the other ones with smaller sample size, it's a lot flatter. But it also might be that maybe the red and the brown are basically have a predisposition to be about the same amount of height. So whatever your proportion maybe doesn't matter as much. And at least with the height prediction, It actually turns out that the ancestries that make the most differences for this are the blue and then the green. So it's a lot of these ancestries, they're not contributing equally to height. If that makes sense. I mean, they all matter for height, but it's like, what are the outlier ancestries when it comes to height? And so really what matters is what's your proportion of those outlier ancestries, okay? And so you can imagine, sometimes that's gonna be informed, sometimes it's not, but it's capturing stuff that maybe you're not gonna be able to get if you're trying to do a traditional GWAS, you're hoping for a great p-value. And in principle, you could combine that with that to do a better prediction of height. I hope a reviewer doesn't ask for that. But that would be a natural next step and say, what if we take this GWAS, what if we take some other piece of information, combine it? And it should work better. But yes, yeah.
So, I mean, I guess on that, when you say this concept of correlation.
Yeah.
It's represented genetically.
So to me, if we didn't see the trend within ancestry, then I would say, if it was just environmental, then everybody that was sampled in this so-called Hansa group, you would expect them to be flat. If it was just environmental.
You think there's no correlation between that? So
Yeah. Well, if there was a correlation with ancestry environment within that group, then there could be something even more complicated. Okay, but
But you think that's not the case?
that's where I'm starting from. There could be something, but you'd have to, but this gets the question, where's the burden of proof, right? So it's like if somebody from the sampling locations in Tanzania, are there known environmental differences for the ones that are more blue ancestry versus the others? That could be looked into. And then that could possibly contribute to this. You're right. And it could be something that maybe it's dietary, right? So like the amount of food that somebody gets at an early age is really going to be affecting body size. You can imagine that that's going to matter. How much does that vary? That could definitely contribute. But I'm more likely to start my null in genetics, but yeah, I think it's still a null.
So what happens if we look at early onset cases?
4 different diseases. Sorry.
For
Yeah.
prostate cancer?
Yeah, so when we do that, so our sample size, we're looking at pretty early onset. So we're not looking at men that are 80 years old. We're looking at men that are 50, 55. And so when we do that, we do see earlier age of onset. In terms of that, I'm going to dodge the question a little bit and look, well, we've looked at early age of onset, we've looked at aggressiveness. And what's a little bit disappointing is when you look at aggressive prostate cancer or early age of onset, you don't get as many GWAS hits. Okay, and they often don't co-localize to the same peaks that we're getting when we look at stuff that's a little bit later. So my work, we've had debates about this in our consortium, but I tend to view it as there are these germline variants that are affecting your incidence rate of prostate cancer and other diseases. Okay, but the ones that are really affecting mortality, those might be secondary mutations that are somatic. Okay, so at least for prostate cancer, you look at the genes, like you look at TCGA, you look at like what are the genes that in somatic cancer evolution that tend to have a lot of prostate cancer hits. Those often are different than the ones that you see in these germline GWAS studies. Okay, but both are associated with prostate cancer. So I tend to think that it's maybe more the incidence, but for the early age of onset, it might be a different cohort, a different set of genes. And we have a moderate sample size for it, but I mean, we don't have that many men that are, younger than 40 or 45 in our clinic. We did have a little bit of imbalance when we were initially doing our study, but we tried to make sure that every study site had about the same age distribution. To get at the age of onset, that actually does matter a little bit when we talk about baldness, because if you have a study where you're looking at how bald somebody was age 55 versus 30 or 45, you can imagine that you might pick up different things in a study where you're looking at it in a different age cohort, right? So like with our MadCat data, we have information of how bald, we've got this, basically it's a survey where you basically have these pictures and basically it's like, okay, how bald were you at age 30? How bald were you at age 45? So there's a recall bias that we added age to covariate to try to fix that. But the other thing is like the vast majority of our men were like, no, I got a full head of hair at age 30. Not everybody. But you can imagine you would get a very different result if you did that. And of course, we had a small number of men that magically grew hair from 30 to 45. We're like, throw them out of the data set because something's weird going on in terms of recall. But my guess is that you would see very different things. We actually see that a little bit when we compare baldness GWAS done in continental Europe versus the UK Biobank. And the UK Biobank cohort is a little bit shifted like 5 years. And so we don't see as good a replication between different European ancestry populations as you might think. And that could be environmental effects, but I think it's just the age that the cohort that's affecting.
So I'm just curious if there's any utility of separating patterns of boldness when it comes to with regards to venture, like different types of boldness might have.
Yeah, that's no, and for the sake of this, we scale things on a 1 to 4 scale. For this, but we've got a full 12-point scale, and also like, is it more like a widow's peak type thing and that sort of thing? We didn't have the sample sizes to really trust going on that scale. And it's because when we did our baldness study, we actually reduced our madcap data set to the ones without cancer. So we're cutting our data set in half. And then with any sort of bin, we have a small number of individuals. I think that would be great. I think we would need much better sample sizes for that. And just people wondering, like, why are we even looking at baldness? One reason for it is that you think about androgen levels might be related to prostate cancer. Well, what we don't know is what were some of these androgen levels throughout their life. But in principle, if you know how bald they were at a younger age, that it's a poor proxy, but at least it's some sort of proxy for androgen levels at a different point in life. And so we're looking to see if there's a compound effect in terms of that. And if there is, it's pretty subtle. It's nothing that's like a. Yeah, we had a second question.
Yeah, the second question, did you calculate variability, for example, on the height GWAS that you trained with this, like the training one, to see how much, like.
Yeah, so with the original one from the Yengo et al. One, so I think Peter Vischer and everybody else that was on this claimed that they were catching all the SNP heritability, but they were catching the SNP heritability in the populations they were looking at. Okay, so one way to think about it is that maybe they were able to catch with a roughly, I always forget if it's 5.4 or 4.5 million, but roughly 5 million individuals. In that cohort, they're doing a good job of catching the SNP heritability, but there are variants that are affecting height that are in other places that just weren't in that study because they were looking at, I don't want to say the wrong, but they're looking at different populations. Okay, so if you think about short stature and Baca, Bacola, Benson populations, that could just be due to population-specific variants that are just not found in other places. And if it's something like that, then that would help explain why this doesn't do as good a job predicting. Okay, and so I think some of it's that you're getting at this, and actually I'm trying to remember from that study of like how much of their heritability was in the low end of the frequency spectrum. But if you get a decent bit of your SNP heritability in the really rare alleles, then that contributes to the fact that you're just going to be missing some of the stuff. So I think, once again, you can interpret this different ways, but I tend to think of this as you've got a lot of SNP heritability in the low end of the spectrum, and then those are more likely to be population specific. But I'm doing a little bit of hand waving that. 5 minutes over, sorry. Right along, just